Hello, everyone. My name is Selena Ferrian. I am a senior scientist in the Department of Pathology at Stanford University. Today, I will give you a brief overview on how to reveal the tumor immunobiology using an imaging technology called multiplex ion beam imaging by time of flight, or MIBI. The first aim of our webinar is focused on how the MIBI is used in cancer research, where I will show you how this imaging technology is applicable to a variety of healthy and neoplastic tissue and what is the outcome that we will be able to achieve. The second aim is focused on learning how to design and optimize a MIBI panel for cancer research. So the MIBI technology is an innovative system which utilizes a continuous primary ion source with orthogonal time-of-flight mass spectrometry to image antibodies tagged with metal reporters at subcellular resolution in formalin-fixed paraffin-embedded human tissue. As you can see from this figure, there are four main instrument components: the sample stage, where the slide with tissue are loaded; the primary ion source and column for sputtering the specimen with primary ions; the electrostatic analyzer, which filters secondary ions with respect to kinetic energy; and finally, the orthogonal acceleration time-of-flight mass spectrometer, which analyzes the quantity of isotope bound to each cell. So to give you a brief overview how this technology works, we have depicted on the right figure the primary ions that are hitting the tissue. And these are the ions, the beam ions, released by the primary ion source. Once the primary ion source or primary ion beam sputters the elemental reporter and tissue endogenous element, secondary ions are released from the tissue. Here we have two types of secondary ions. We have monatomic and polyatomic ions. And what does monatomic and polyatomic ions mean? Monatomic ions are elemental ions or single atoms with high energy, which move faster. They are the ones that go through the TOF, where they get sorted by their mass and they hit the detector, while polyatomic ions are molecular ions clustered together with low energy, which move slower. Basically, they are those ions that get stuck and do not reach the top because of their low energy. So to be more specific, they are stuff that comes out from the tissue, clumps of atoms which covalent bonds have not been broken, for example, because no much energy has been transferred to them, anything that has not been completely ionized, or maybe atoms that make adapt with organic material such as carbon, hydrogen, oxygen, or molecular conglomerates, etc. So the question is, how does the filtering is done? I won't go through all the details, but basically there is an energy filter in place by the electrostatic analyzer, where the gap is chosen to maximize the signal to noise. By gap, I mean the peak where the signal and noise almost disappear. So the idea is to break all the covalent bonds and have monatomic ions going through the detector. Moving forward, these monatomic ions enter the orthogonal time-of-flight mass analyzer, which analyzes the quantity of isotope bound to each cell. The ions are then separated based on their masses and are accelerated to the detector. Finally, the detector measures the quantity of each isotope for each clone. The next point is to show how MIBI can simultaneously quantify all naturally occurring elements from hydrogen to uranium. We use these capabilities to simultaneously provide the subcellular expression and localization of up to 42 elementally labeled antibodies, as well as native biological elements such as phosphate and iron. We additionally, as you can see on the next figure, we additionally show that MIBI is designed to support large field of view with low noise. As you can see in this figure on the left, which we call survey scan, usually done at low resolution to capture, for example, the tumor heterogeneity. And also, As you can see on the next set of images on the right, it allows for repeat scanning of a single section, for example, a specific region of interest identified during the survey scan. In this context, region 2, 6, and 7 of breast cancer tissue, enabling study design at different scales of throughput and resolution, or even acquisition of multiplex three-dimensional images. The next slide is showing the acquisition time in minutes and the resolution achieved for a 200 micron square field. As you can see from the diagram, low resolution 1 micron takes only 1 second to be scanned, while high resolution 260 nanometers take approximately 1 hour. Therefore, as reporting on the right figure, we can proceed with a survey scan followed by a rescan of the same field of view on high resolution to unveil further detail, for example, subcellular localization of certain targets, like laminin and double-strand DNA in this example. I would like also to highlight that currently the next generation ion source commercially available is capable to provide much brighter imaging 4 to 5 times faster, meaning that in approximately 18 minutes, you can have a 400 micron square image scan on high resolution. Moving to the next slide, we can see that we're currently implementing MIBI on different biological tissue, like liver, placenta, lung, upper and lower GI, breast, spleen, lymph node, skin, tonsil, and also in brain, in order to characterize the host immune responses within and around the tumor microenvironment, with a particular focus on the stimulatory and inhibitory immunocheckpoint molecules and their ligands. We currently work with both SIPE as well as frozen tissue, mainly from humans, monkeys, and mice. Let's now provide some example on how our lab has implemented MIBI technology in the field of cancer, inflammation, and infectious diseases. Much like a diamond disperses light into its individual wavelengths, this technology allowed us to separate and more specifically evaluate the different features underlying the state of tissue in a multitude of diseases. Therefore, identifying epithelia and mesenchymal component, immune cells, vascularization, tumor-specific target, functional molecules, and transcription factors. And what type of information are we able to extrapolate? What other type? Using this method, we are able to extrapolate from a single biopsy not only information for 42 targets analyzed simultaneously, but also their localization and spatial arrangement around and within the tumor microenvironment. So, as you can see from this figure, we are currently working on several cancer types depicted in blue, like triple breast negative cancer, glioblastoma, several carcinomas in lung, pancreas, bladder, and kidney, melanoma, and breast carcinoma. We are also actively working in other inflammatory and infectious diseases like Alzheimer's, pulmonary hypertension, preeclampsia, and mycobacterial tuberculosis. On the bottom of this diamond, which I depicted in red, you can see how we are implementing MIBI for immunomonitoring clinical trial in cancer and also investigating cancer treatment toxicity, for which I can show you right now an example. This is a recently submitted publication in collaboration with UCLA, where we reported implementation of the MIBI technology to biopsy from a patient which developed treatment toxicity. The patient was found to have colorectal carcinoma as well as endometrial carcinoma. After receiving combination of chemotherapy and brachytherapy treatment, CT-guided biopsy showed a metastatic lymph node. The patient sought consultation at UCLA, receiving treatment for nivolumab and achieving complete response at 17 months. However, after 32 months of therapy with nivolumab, the patient developed severe epigastric pain and nausea and anorexia. On these 4 representative images selected, the biopsy from the gastric antrum and body show similar features which were characterized by diffuse eroding mucosa with crypt entry and dropout associated with intraepithelial and lamina propria mixed inflammation that included frequent lymphocyte and neutrophils. We therefore analyzed the gastric specimen using an immunopanel comprising targets for T cells, B cells, regulatory T cells, NKs, and myeloid components. We also introduced targets to identify the non-immune compartment, such as endothelial cells, mesenchymal and epithelial cells, and finally, a few functional targets. As you can see, on this image, we routinely introduce all the necessary tissue control to ensure the target sensitivity and specificity. Here, we present a case of nivolumab-associated autoimmune gastritis. To explain a possible role of the main cell type offenders in this dramatic clinical response, we selected 13 representative regions of interest from the HIM staining on the left of the gastric biopsy. And for this purpose, we leverage the ability of MIBI to identify the presence and proportion of infiltrating immune cells from a single section of biopsy specimen. Our analysis revealed a gastritis characterized by significant immune infiltration noted primarily in the lamina propria, mainly composed of T cells and to a lesser extent by macrophages and mast cells. Sparse B cells and dendritic cells and NK cells were also noted in the lamina propria. In addition, we found moderate levels of CD31 cells in the lamina propria, highlighting a significant degree of vascularity across the gastric mucosa. On the other hand, epithelial cells, highlighted in yellow on the first figure on the left, displayed consistently elevated interferon gamma expression throughout the region, which may be a potential contributor to the observed gastritis. The majority of T cells were CD4 and expressed CD45RO and HLA-DR antigens. We also observed FOXP3 regulatory T cell diminished, consistent with the pro-inflammatory phenotype observed here. On the other hand, the majority of CD8 T cells lacked granenzyme B, while the macrophage-infiltrated areas surrounding gastric epithelium lacked HLA-DR and interferon gamma. Quantitative analysis performed on all the regions further corroborate that CD45 positive cells, as you can see on the diagram below, represent approximately 40% of the cells found in the gastric tissue, followed by mesenchymal, epithelial, and endothelial cells. In addition, the majority of the immune cells, approximately 70%, were T lymphocytes, followed by macrophages and mast cells, while the remaining immune cell subset each accounted for less than 5%. So taken together, the striking feature in this case study of PD-1-associated gastritis include lamina propria expansion by mononuclear inflammation characterized by CD4 and CD8 T cell infiltrates, lacking or with reduced expression of granenzyme B and FOXP3 respectively. Epithelial injury from crypt apoptosis, intraepithelial lymphocytic and neutrophilic inflammation, as well as expression of interferon gamma by epithelial cells. To conclude, we were able to investigate several features via MIBI-TOF, which highlighted the need of additional investigation to increase the understanding of the regulation of inflammation and epithelial cell changes in the upper gastrointestinal tract after PD-1 therapy. The next slide briefly highlights a work in progress in collaboration with the Rabinovich Lab here at Stanford University, where we aim to profile dendritic cell population in pulmonary hypertension patient biopsy. The analytic validation was carried out on human FFPE tissue where representative 2 mm core of tonsil, spleen, lymph node, GI, placenta, normal lung, and diseased lung tissue were compiled into a tissue macroarray to optimize and validate the proposed 38 marker panel. The patient cohort and tissue control were stained simultaneously and analyzed by MIBI. The final testing is performed and evaluated on the same TMA by single-plex IHC, where the antibody that show expected results across different tissue by immunohistochemistry were compared to the MIBI expression pattern in combination with visual comparison of marker co-expression. And the last example is another work in progress in glioblastoma multiforme, which started as an internal project and has recently been co-founded by Parker Institute for Cancer Immunotherapy and the Cancer Research Institute. The overall aim of our project is to accurately determine the infiltrating and resident myeloid cell types and state in glioblastoma biopsy, and to map specially resolved cellular networks. We want to understand how cell-cell communication changes the information processing and ultimately affect tumor development and treatment. To this end, we are using these novel experimental and computational approaches that allow for highly multiplexed, spatially resolved, comprehensive single-cell network analysis of cells in brain tissue. Using our MIBI technology, we will be able to unveil the role of glioma infiltrating macrophages in disease pathogenesis. More specifically, we hope to elucidate which cell phenotypes can predict patient survival and response to treatment, how they behave, and where they are located within the brain microenvironment. These, hopefully, will give us insight for future immunotherapy strategy in glioblastoma. And here below is simply display how MIBI validation has been carried out for this project, where appropriate tissue control like tonsil, spleen, placenta, liver, normal and neoplastic brain tissue were included to assess specificity and sensitivity for each target included in the panel. So after showing how we apply the MIBI technology in the field of cancer, the second part of our webinar will focus on how to design and optimize a MIBI panel for cancer research. In order to construct the design MIBI panel, the following workflow that I will display on the next slide. The following workflow for the initial quality control procedure for each reagent is routinely carried out in our lab. The analytic validation is carried out on human formalin-fixed paraffin-embedded tissue microarray. Antibody purity is evaluated before conjugation by SDS page in reducing condition in order to make sure that the antibody does not contain any carrier that interfere with the metal conjugation. After successful metal labeling, conjugates are tested and titrated by single-plex chromogenic IHC and subsequently by MIBI on the same tissue. New clones are tested before and after metal conjugation to ensure that metal labeling of the antibodies does not interfere with their target specificity. For each metal conjugated antibody, the working titer is determined by MIBI as the titer that yielded maximal separation between signal and noise across different types of tissue. Tissue processing and staining and quantitative analysis are carried out as described in our recent publication in Cell. Moving forward, after the initial quality control procedure, we can proceed with constructing the metal-labeled antibody panel. And how does this is done? We first assign the appropriate metal to the antigen such that the proposed markers will accurately define each population of interest. A few considerations should be taken into account during this step. First, we need to choose the right combination of metal antigen according to the nature of the tissue study. meaning that if you are characterizing the immune infiltration metastatic lymph node, you would want to conjugate CD45 with the dimmest metal to avoid potential +1 spillover. And here is reported for your convenience a table summarizing the relative sensitivity between elemental metal tags. And just to give you a brief explanation, in order to estimate the effective sensitivity for detecting metal tagged antibody in tissue, our group stained serial section of tonsil with 16 different elemental metal isotopes conjugated to an identical monoclonal CD45 antibody. From each section, the 100 micron field of view was imaged by MIBI. Following imaging, the remaining tissue was dissolved and run in solution phase elemental ICP mass, from which the elemental isotope concentration was extracted and calculated. Sensitivity was calculated by comparing the total number of metal atoms, the 30 mm, and the absolute solution quantification. And finally, values were normalized to the tissue area used for both measurements. As you can see from this figure, the sensitivity of the MIBI exceeded by up to 37-fold that of the most sensitive element 159 Tb reported using a mass cytometry, which is represented by the last histogram bar. On the next slide, we are highlighting that we need to remember that certain metals like PR and ND or praseodymium and neodymium may result in +16 spillover due to the oxidation, which can be easily avoided by assigning the right antigen in this channel. Next step, to construct successfully a MIBI panel is the tissue macroarray design or TMA design. Testing appropriate tissue set to determine analytic sensitivity and specificity for each target introduced in the panel is part of our MIBI panel validation. Based on the target you wish to introduce in the panel, you will have to design a different TMA containing both negative and positive tissue control. Let's now provide an example. I plan to characterize using a 40-marker panel the immune infiltration breast carcinoma tissue with a specific focus on the myeloid component such as dendritic cells and macrophages. For this purpose, I will design a TMA containing the following tissue type: tonsil, placenta, liver, and spleen in order to optimize and validate the desired MIBI panel. I would also, whenever possible, include healthy brain tissue as it represents an optimal negative control for many lymphoid markers. Another aspect to take into account when selecting appropriate tissue types to validate a desired MIBI panel is that often during chronic inflammation like cancer, receptors can be upregulated or downregulated. Therefore, I would strongly recommend to include in your TMA design also normal breast and breast carcinoma specimens in order to see first how certain markers behave during inflammation and second, how to adjust your titration according to the tissue of interest, which in this example would be breast carcinoma. Once the TMA is designed, as you can see on the next slide, the overall tissue quality will be evaluated on a serial hematoxylin and eosin section, or HNE section, under light microscope. This initial section, representing our reference standard for histological examination, should be followed by 6 serial sections on MIBI slide, or GO slide, as you can see on the next slide, in order to generate 6 titers of the panel. The working titer is determined as the titer that yields maximum separation between signal and noise across tissue, as you can see depicted in the bottom of this figure. This approach will help to assess the specificity and sensitivity for each target with regard to its cellular localization, subcellular localization, therefore nuclear, membranous, and cytoplasmic, as well as co-expression of proteins across different tissue types. The expected staining pattern should be noted for reaction comprising of neoplastic and non-neoplastic tissue control. When a negative tissue control is unavailable, appropriate ROIs or region of interest with both positive and negative areas will be evaluated. Once the antibody cocktail is generated, a single core of tonsil, as you can see on the next slide, a single core of tonsil included in the same slide of the sample will be stained during each experiment and run for longitudinal standardization. Data emerging from this internal control will be examined for change in signal intensity of the positive populations. In parallel, a TMA section will be stained with a sample of interest or cohort in order to provide further quality control of assay performance or other potential issues emerge during the staining. To conclude, the evaluation criteria for the antibody panel is performed by comparing the MIBI scope expression pattern to the conventional single-plex chromogenic IHC. An example on how the comparison of selected markers carried out is reported in our recent publication in Cell, where we show how the expression pattern in tissue for each marker analyzed was equivalent in both single-plex chromogenic IHC and MIBI samples. As you can see from this figure, the cell morphology and cellular localization of the study markers is comparable between the two assays. For example, CD20 was primarily found in the germinal center, where B cells are the predominant population, while CD3 and CD4 primarily stain cells in the interfollicular zone, which is populated with T cells. As a subcellular localization of the transcription factor Ki67 was predominantly nuclear, whereas CD4 and CD20 localized in the membrane, as expected. To conclude, in combination with visual comparison between IHC and MIBI, we also evaluate the co-expression of protein on the same cell based on known biological property of the marker or no significant co-expression due to expression in a different compartment of the cell or on a different cell type.